Hi everyone, my name is Alex Levitayo. I'm a junior at Marian University, and today I'm joined here by my lab partner.
I'm Erica Sheagley, and I am also a junior at Marian University.
And today we'll be presenting to you about our research that we've been working on this past year, which is determining the effects of pharmaceutical contaminated irrigation water on Arabidopsis thaliana plant stress responses. So for some background and some significance about our project, we have found that many papers have found that pharmaceuticals are usually found in treated wastewater, even though they're treated. And a lot of times many countries use treated wastewater to irrigate the plants and their crops. So what we were interested to find out was the connection between watering the crops and plants that we consume as food from treated wastewater, which could have pharmaceuticals in them. So for our project, what we wanted to do was focus on one pharmaceutical drug and see how that affected a plant. So we chose to do our project over metformin, and metformin is one of the most prescribed drugs in the world for type 2 diabetes. We know that it has been found in treated wastewater, and we know that it can be uptaken by the plant through the roots. And then we're also going to treat the plants, which are the Arabidopsis thaliana plants, to metformin water solution and see if the plants have any stress responses.
So for some of our rationale and significance, we're going to be looking at how this drug influence on plant growth and plant survival is. So currently, there are no actual quality standards with regards to pharmaceutical concentration in reclaimed waste irrigation water. Metformin, which has been detected in wastewater treatment plants, influence and effluent at concentrations ranging from respectively 101 to 129 and 2.2 to 29 micrograms per liter. So treated wastewater is used to irrigate crops. However, current water purification machines are not able to eliminate all the pharmaceuticals. Therefore, many pharmaceutical residues are still bound to remain. So this goes within our improperly disposed of drugs, which could potentially lead to various effects in the plants in our environment.
For our project, what we will be doing is we're going to be growing our Arabidopsis thaliana plants for 2 weeks, and then they'll be synchronized while they're growing. And then once they're grown to 2 weeks, we're going to expose them to different concentrations of a metformin water solution and see how they respond to those different metformin water solutions. And then we're also going to, or we also, wait, we're also going to do like a chlorophyll mass spec assay as well, which is going to measure the concentration of chlorophyll before and after the treatment. So to assess plant stress response, we're going to be looking at the endoplasmic reticulum induced stress response, which causes the UPR by measuring BIP protein expression through qPCR analysis. So this figure over here just shows that in one paper that we found, metformin does induce ER stress in cancer cells. And then, so what happens during ER stress is the unfolded protein response turns on because proteins start to accumulate and unfold. And one of the proteins that was found that is turned on with metformin is the BIP protein, and BIP is a chaperone protein that helps alleviate the ER stress, and it turns on when the UPR is turned on. So we know that metformin does induce stress and turns on BIP in cancer cells when they are exposed to metformin. So that's our rationale behind exposing metformin to plants, because there haven't been a lot of research and findings found relating metformin on plant stress responses. So we're going to do that and then expose them to 4 different concentrations with one control, which is just plain water. And then we're going to measure the stress response through qPCR.
And then for our experiment 2, we are also going to be doing a chlorophyll analysis. So that'll be utilized to monitor, predict, and examine stress in plants. So since chlorophyll content actually changes based on environmental stress, it is also known that chloroplasts in plant work and behave similar to how mitochondria do as well. So this similarity between plant chloroplasts and mitochondria will be critical in characterizing the metformin stress response on these plants. So we'll be using a mass spectrometer, which is actually an incredible accurate piece of instrumentation that we'll be able to use to determine this abundance of elements and compounds. So right now, the interactions of metformin on plant chlorophyll content is currently unknown. So we'll be looking into that to see exactly what ends up occurring within our actual experiment.
So one of the first steps that we did was we first plated our seeds to grow them, and then we plated them so that they would grow synchronized at the same rate, so that by the time that we water them, they'll be at the same age and that they'll be all the same. So what we did was we sterilized the seeds, and then we plated them onto an agar plate, and then we allowed the seeds to grow in a growth chamber for 1 to 2 weeks. And in this picture here just shows how our little seedlings grew after 2 weeks. And then they were also placed into our growth chamber here.
So then once the 1 to 2 weeks were up, we transferred these seeds to soil and then grew and watered these synchronized plants for 2 weeks. And this was just like normal water. There wasn't anything in it or anything. This was just to kind of start out these plants so that they could get to about 2 weeks of full growth. And then we could actually start to do our experiment and see how that treatment ends up going on.
And then this picture here just shows what the plants look like after watering the plants for 2 weeks with just plain water. So this is right before we started our watering experiment with the different concentrations of metformin. So as you can see, all the plants look happy and healthy, and they're all watered with water. So now in this slide here, we divided up all the plants into different Tupperware, which has different concentrations of water of metformin. And so you can see here that we have a 100 nanomolar solution, a 10 micromolar solution, a 1 millimolar solution, and a 900 nanomolar solution, and as well as our control. So for the next few slides, we're going to see a progression of the different plants and how they react to the different concentrations. So this is the first day that we watered them. So we don't really see much change compared to like a few days before.
And then by March 4th, we ended up seeing quite a noticeable difference within our different groups. So you can see here on the left, we have our control that looks healthy, happy, growing really well. And then we start to get to our 100 nanomolar, and we start to see some actual discoloration. We're seeing some yellowing, and also the plants are actually beginning to kind of wilt a little bit. Our 900 nanomolar is also, once again, we kind of see some of the plants are starting to yellow a little bit, and there is also more discoloration. They're kind of starting to die a little bit more, probably the most that we're seeing here. And then our 10 micromolar seem to be doing really well, actually. They're really big for the majority of the plants overall. And then our 1 millimolar as well is kind of in the middle, I'd say. There's a decent amount that is still growing, and we're not really seeing much discoloration. The majority of the leaves are green here.
And then on March 15th, a few days after, we can see that our 100 nanomolar concentration group, like you can see that the plants are basically dead. There's not that much green. It looks like they all wilted away. And that's our smallest concentration of metformin. Next, we have our second smallest concentration, which is the 900 nanomolar. The plants are also turning yellow and starting to wilt. But then as you keep getting higher with the concentration, you can see that these plants over here are still green. But then you can see some yellowing on them as well. But our control plants are still green and growing as well. So you can see how the plants react to different concentrations of metformin. There might be a bad response when they are exposed to a smaller concentration, such as our smallest one, which is the 100 nanomolar. And they might also have another stress response to the highest concentration, which is 1 millimolar. Like you can see some yellowing in the plants, but they're still green and growing, not as bad as the 100 nanomolar. So what we're thinking here is that there might be a little threshold for the plants depending on their stress when exposed to the different concentrations.
So what's going to end up happening next within the next couple weeks is we are pretty much finished watering with our plants. We are conducting qPCR, and we're going to be able to, the plan is to measure the BIP expression. And we'll be doing this through 12 different tubes that we have, with taking 3 leaves from each of the groups, from each plant. And then we'll be able to do an analysis on that and then see how things end up going on. And then we'll also be following up with our second experiment as well. So that will be our chlorophyll concentration. So we'll be taking a look to see exactly what we find, what our results are, and we will be updating that as well in the future.
Then lastly, we would just like to acknowledge all the help and support that we received from Sigma Zeta, especially for funding us and providing us with our grant. And then we also want to thank our biochemistry professor, Dr. Collins, as she has been mentoring us through our project and guiding us as well. So thank you very much.
Yes, thank you.